As we age, our immune system undergoes significant changes, particularly in the function and composition of T cells. These changes contribute to increased vulnerability to infections and reduced vaccine effectiveness in older adults. One key aspect of this process is the accumulation of regulatory T cells or TREGs, which can suppress protective immune responses. But what drives these changes at the cellular level? Recent research suggests that compromised DNA repair mechanisms may play a pivotal role in accelerating T cell aging, leading to both functional decline and altered immune regulation. DNA damage naturally accumulates in our cells over time. Normally, our bodies have robust DNA repair systems to fix this damage. However, as we age or if these repair systems are genetically compromised, DNA damage can build up. This accumulation is now recognized as a central driver of aging and is linked to many age-related diseases. In immune cells, especially T cells, this can lead to changes in their ability to respond to threats and maintain a healthy immune balance. One of the key players in DNA repair is the ERK1 gene. It helps fix various types of DNA damage, including those that distort the DNA helix or block transcription. Mice with mutations in the ERK1 gene, known as ERK1-delta-7 mice, have impaired DNA repair and show signs of accelerated aging. These mice provide a valuable model for studying how compromised DNA repair affects T cell function and aging. To explore the effects of DNA repair deficiency, researchers compared young and aged wild-type mice with ERK1-delta-7 mice. These genetically engineered mice exhibit rapid aging due to their inability to repair DNA efficiently. By analyzing their immune cells, scientists can observe how DNA repair impacts T cell populations and function, offering insights into the aging process at a molecular level. T cells are a diverse group including helper T cells, cytotoxic T cells, and regulatory T cells, TREGs. With age, the total number of T cells tends to decrease, but the proportion of memory T cells increases. Memory T cells are those that have previously encountered antigens and can respond more rapidly upon re-exposure. In both aged wild-type and ERK1-delta-7 mice, researchers observed a rise in memory T cells, suggesting that DNA repair deficiency mimics some aspects of natural aging. A hallmark of immune aging is the accumulation of regulatory T cells or TREGs. These cells help maintain immune tolerance and prevent autoimmunity, but in excess they can suppress beneficial immune responses. Both aged wild-type and ERK1-delta-7 mice showed increased frequencies of TREGs, particularly within the naive T cell compartment. This suggests that compromised DNA repair drives the accumulation of TREGs, contributing to the immune suppression seen in aging. Not only do TREGs accumulate, but their phenotype also changes with age. In ERK1-delta-7 mice, TREGs showed reduced expression of CD25 and increased expression of PD1, markers associated with an aging-related, less responsive state. These changes mirror those seen in naturally aged mice, indicating that DNA repair deficiency can induce an aging-like phenotype in TREGs, potentially reducing their effectiveness in immune regulation. Interestingly, while naive TREGs accumulate with DNA repair deficiency, the frequency of memory TREGs does not significantly change in ERK1-delta-7 mice compared to controls. This suggests that the accumulation of TREGs with age is primarily driven by changes in the naive T cell pool, rather than expansion of memory TREGs. The distinction between naive and memory TREGs is important for understanding how immune regulation shifts with age and DNA repair status. Helper T cells, or Th cells, play a crucial role in orchestrating immune responses. In ERK1-delta-7 mice, the phenotype of naive Th cells was largely unchanged, but memory Th cells showed increased PD1 expression and decreased CD122, markers associated with aging. This indicates that DNA repair deficiency selectively impacts certain T cell subsets, particularly those involved in memory and regulation. Cytotoxic T cells, or TC cells, are responsible for killing infected or cancerous cells. In ERK1-delta-7 mice, naive TC cells were largely unaffected, but memory TC cells showed increased PD1 and decreased CD122, similar to changes seen in memory Th cells. These findings suggest that compromised DNA repair primarily affects the memory compartment of T cells, potentially reducing their ability to respond to new threats. The mTOR pathway is a central regulator of cell growth and metabolism and has been implicated in aging. Inhibiting mTOR with the drug rapamycin has been shown to extend lifespan and improve some aspects of immune function in mice. Researchers hypothesized that blocking mTOR might slow or reverse the T cell changes induced by DNA repair deficiency. To test this idea, ERK1-delta-7 mice were fed a diet containing rapamycin. The results were intriguing. While rapamycin reduced the proportion of memory T cells, it did not decrease the overall accumulation of TREGs. Instead, it increased the proportion of memory TREGs with high CD25 and PD1 expression. This suggests that mTOR inhibition can shift T cell populations, but may not fully counteract the effects of DNA repair deficiency. Aging and DNA repair deficiency both lead to reduced T cell responsiveness. In experiments, T cells from ERK1-delta-7 mice showed diminished proliferation and lower upregulation of the activation marker CD25 in response to stimulation. This mirrors the decline seen in aged wild-type mice, indicating that DNA repair is crucial for maintaining T cell function as we age. Why do TREGs accumulate when DNA repair is compromised? One theory is that DNA damage in the microenvironment leads to the buildup of senescent cells, which secrete inflammatory factors like IL-6. This pro-inflammatory environment may drive the expansion of TREGs as a compensatory mechanism to control inflammation, but at the cost of suppressing protective immunity. The accumulation of TREGs and reduced T cell responsiveness have real-world consequences. Older individuals or those with compromised DNA repair may be less able to fight infections or respond to vaccines. Understanding these mechanisms is vital for developing strategies to boost immune protection in aging populations. While these findings shed light on the role of DNA repair in immune aging, there are limitations. The ERK1-delta-7 mouse model does not cover all DNA repair pathways, and it's unclear whether the observed changes are due to intrinsic T cell defects or the surrounding microenvironment. Additionally, the effects of different doses of rapamycin and their impact on various immune cell subsets warrant further investigation. Future research should explore how different DNA repair pathways contribute to immune aging and whether interventions like rapamycin can be optimized to restore immune function. Expanding the range of markers and cell types studied will help clarify the complex interplay between DNA repair, aging, and immunity. Understanding the link between DNA repair and immune aging could inform new therapies for age-related immune decline. Targeting DNA repair pathways or modulating TREG accumulation may offer ways to enhance vaccine responses and reduce infection risk in the elderly. In summary, compromised DNA repair accelerates the accumulation of regulatory T cells with an aging-related phenotype and reduces T cell responsiveness. While mTOR inhibition with rapamycin can shift some T cell populations, it does not fully reverse these changes. These insights highlight the importance of DNA repair in maintaining immune health as we age. To better understand these findings, researchers used advanced techniques like flow cytometry and dimensionality reduction to map T cell populations. These methods reveal subtle shifts in cell phenotypes and help pinpoint how DNA repair impacts immune aging at the single-cell level. The study of DNA repair and immune aging is opening new avenues for understanding how our bodies defend against disease as we grow older. By unraveling the molecular mechanisms behind T cell aging, scientists hope to develop interventions that preserve immune function and improve health span. Continued research in this area promises to bring us closer to healthier aging for all.